Good evening, ladies and gentlemen, and a warm welcome to this Francis Crick lecture by Gregory Jefferis from the University of Cambridge. My name is Gero Miesenberg from the University of Oxford, and as Greg's nominator and admirer, I have the pleasure of introducing him and the lecture tonight. Before I say a few words about Greg and the Crick lecture, I was told I have to mention several housekeeping issues. First, I should remind you to switch all mobiles to silent. Second, I should tell you that we would very much appreciate feedback on the lecture, and you can give that feedback by scanning the QR code on the back of your badges, and that takes you then to an online feedback form. There are no planned fire evacuations tonight, luckily. But should the fire alarm sound in case of a real emergency, please vacate the building via the marked exits. There's obviously one in the back and exits in the front. And then assemble at the assembly point, which is the top of the Duke of York steps to the right of the building as you exit. Finally, the event is being webcast live tonight and recorded for posterity, and it will be supported by live speech-to-text translation. So you'll see some words that have nothing to do with Greg's slide design flashing on the screens. The Crick lecture was endowed in memory of his long-term friend and collaborator Francis Crick by Sidney Brenner in 2003. When I nominated Greg, there was an age restriction on the lecture. The recipient at the time of delivery had to be below 40 years of age. So from that you can tell that Greg, despite his significant accomplishments, is still quite young. The lecture is open to all areas of biological sciences, but with a special emphasis on those that were of particular interest to Francis Crick. And these areas include genetics, molecular biology, neuroscience, structural biology, and fundamental theory. Now, Greg, I think, straddles two of these areas because he could be called a structural neurobiologist. He's an incredibly creative neuroanatomist who deciphers the structure of brain circuits with the intent of discovering the physical substrate of innate and learned behaviors. Much of his work is focused on how odorous molecules, through the distributed patterns of receptor activation that they elicit in the nose of fruit flies, then inform perception, choice, and action. And that's what we're going to hear about tonight. Greg was an undergrad at the University of Cambridge and then moved to Stanford for his PhD with Leach and Luo. He emerged so fully formed from the womb of graduate school that he skipped a formal postdoc and began an independent career as a research fellow in the Department of Zoology at Cambridge, and switched later on, after a few years, to the Laboratory of Molecular Biology, also in Cambridge, where he is now a tenured group leader in the neurobiology division. Please join me in welcoming Greg.
Thank you very much indeed for that very generous introduction, Gera. I'm extremely grateful to Gera and to other senior colleagues who nominated me to give this lecture. And I'm also grateful to all of you for coming along and standing in the rain, some of you, to do so. I hope that I'm going to fulfill what is promised this evening. So there are many people really that I should thank for the chance that I have to stand in front of you today. And I actually thought that I would do that a little bit more precisely by looking at all of the co-authors of the papers that I've written. And right now it stands at 187 people. Each of these people who are named here has contributed something to what you're about to hear. Unfortunately, I can't pick everyone out, but that big name in the middle there, 10 papers, is my PhD supervisor, Li Chin-Lo, and I would like just to acknowledge his contribution to my scientific journey. So just so that we sort of know what's coming, a brief menu. So I'm going to give what I hope is a reasonably gentle introduction to brains, neurons, and flies. And then I'm going to tell you a first scientific story, and that's sort of the love/smell part of the title. And that's about wiring differences in male and female brains. Then I'm going to have a brief interlude, which is a little bit more technical, and that's going to talk about connectomics, which is both an old and a new science for mapping the brain. And then the second scientific story will be about memory and smell, so learned and innate behavior. So with all of that out of the way, I'm going to start with a question. And this is a question for the audience. So what's this? Any ideas? No? So I'll give you a clue. It comes from the British Museum. It's from the Egyptian Gallery. Anyone from the audience? Shout out if you know. Exactly. So this was used during the process of mummification for removing the sort of semi-liquid brain from the unfortunate deceased. Now, of course, the ancients were really too sure what was the seat of the mind, and the Egyptians took special care to preserve the heart rather than the brain because they thought that was the most important thing for the afterlife. Now, of course, we know that it's not the heart, but the brain which is the seat of the mind. And I can actually remember quite vividly the first time that I held a human brain in my hands. It was when I was a PhD student. And it was sort of a really moving experience, right? Because somebody had donated this precious organ for research and education. And yet there was also a sort of niggling sense of disappointment, because when you looked at it, it was hard to imagine how could this object encode all of our feelings, our desires, our memories, this sort of, liter and a half of slightly squishy gray stuff. But actually, it was at a pretty similar moment that I started to look at the structure of individual neurons inside brains. And I think that was really when neuroscience completely gripped me, because understanding how these beautiful individual nerve cells contribute to all of these behaviors has really been the focus of my work over the last 15 or 20 years. And I hope I can share you some of the excitement that I've felt for that. So I don't actually work on the human brain. I hope nobody's disappointed by that. I don't even work on the mouse brain, which is shown roughly to scale here. What I work on is the fly brain. So I guess those of you at the front can probably see that. Maybe others can't. And there are various reasons for that. One of them is just about numbers. So in the human brain, there are about 100 billion neurons. In the mouse brain, about 100 million. And in the fly brain, 100,000. So that's actually a countable number, and many of these neurons are my friends. I know them by name, I can recognize them. And that process is ongoing. Now, let's actually zoom into that fly brain. So there it is. This is a poppy seed, if you haven't seen one close up before. So it's a little bit less than a millimeter across, okay? And this is the sort of stuff that's inside the fly brain. So here you have a collection of about 10,000 neurons, about 10% of what's in the fly brain. And so as a neuroscientist, essentially, my job is to think about this sort of computational device that takes information in and turns it into behavior, and to try and figure out how does that happen. And at the moment, that really resolves to probably two key questions. How are all these neurons, these nerve cells, connected together? And how does this let them work together to solve complex problems? And one of the key approaches that we use is the combination of genetics and molecular biology, which does, as Gary indicated in his introduction, at least hark a bit back, both to Crick and to Brenner. So some of you have probably heard of Drosophila, properly called the vinegar fly rather than the fruit fly, and maybe know that it's a common genetic model organism, that people use it for genetic experiments. But it's also used for neurobiology now, and there are various reasons for that, which I hope will become apparent through the talk. But one of them, which I think for me is quite motivating, is simply that the behavioral complexity of this animal, which is more and more surprising the closer you look, is actually quite high compared with the number of neurons that the brain actually contains. So I'm going to show you what, at first glance, seems a rather simple behavior. So it's a visual escape behavior filmed by my colleague Gwyneth Card. So she sort of plays a sort of virtual fly swat at the fly, and then it's being filmed, and here you can see it's slowed down 50 times its response. So what you should do is you should watch the legs here, this pair of legs in the middle of the fly, and see what she does with them. So she's moved them forwards so that she can jump backwards away from the approaching visual stimulus. And this was done in milliseconds. And this is a flexible behavior on top of something that you might think is just extremely hardwired and sort of, coarse, escaping from an approaching object. Now, I'm not going to talk about escape behaviors. What I'm going to focus on initially is sexually dimorphic behaviors. So that is the kind of behaviors that are essential in any species where there are two sexes who need to find each other and convince each other that they might want to mate and reproduce. So I could have chosen a few different examples, but I'm going to choose actually not the flies in the middle here, but this pair of spiders here to illustrate some of the complexities and actually the beauty of here simple invertebrate behavior. So here we have boy meets girl, peacock spider style. And you'll see soon where he gets his name. There's the female. The male is approaching gingerly, waving his legs. There's his tail. You can see how he gets his name. Isn't it splendid? So I'm not sure what she thought. If she was in a good mood, then maybe they mated afterwards. If she wasn't, then maybe she ate him, or maybe she did both. But I do think there's an enormous amount of interesting stuff that's encoded in this little simple interaction, right? Because there's the body of this male spider, this beautiful pattern, which is species-specific. And there's his behavior, which is also species-specific and controlled by his brain. And then there's the female who's watching all of this and deciding whether it, meets the bar. And again, she's looking at this, and these are species-specific behaviors expressed by this other animal. And so all of that somehow is encoded in the genome, as well as the structures of these two brains. And I think that's amazing. And someday I'd love to understand how that happens. Now, we actually work with a slightly more prosaic model system, so sexual behavior in the fly. But when you look closely, there's actually a lot going on. So obviously, there's actually an extended courtship step. The male there is actually sticking out his wing to sing to the female. Eventually, there might be copulation and then, of course, egg laying afterwards. But there are other behaviors that might surprise you. So here, for example, is male-on-male aggression. And they do this when there's something to compete for. So, for example, females or food. It's basically like stags, right? But just in miniature. So let's look a little bit more at this courtship step. This is essentially a female decision-making process. She has to decide whether she's going to mate or reject this male. And she does so on the basis of a wide range of sensory information. So there's smell and taste and vision and song, all sorts of things. And she's integrating all of these. But I'm going to focus on the sense of smell for the next little bit. So in particular, there are pheromones, so smells produced by one animal to signal to another. And the one that we've worked on most is this pheromone CVA, cis-vaccinyl acetate, produced by male flies, which has this rather remarkable property that the same molecule can produce different behavioral responses depending on who's smelling it. So if you're a male fly, this makes you want to reject or even makes you more aggressive. Whereas if you're a female fly, this makes you more likely to mate. Okay? And so we'd love to understand what is the basis in the brain of that difference in perception and eventual behavior. And so our entry point was genetics, actually. So here's a wild-type male and female. You can see the male sticking out his wing to sing and chasing after this female. And here are some mutant animals, mutant males, actually. So even those of you who are not experts in fly behavior can probably see that something slightly curious is going on there. There's this sort of conga dance where the male flies are chasing after the one in front and being courted by the one behind. So generally speaking, that doesn't happen with wild-type flies. This is a property of this particular set of mutants. And so what is then this fruitless gene, and why does it produce this change in behavior? So I'm going to summarize in a very little space a huge amount of work from a number of fly labs. But the bottom line is that this fruitless gene is a transcription factor, so that's a gene which controls the expression of other genes. And it's expressed in about 5% of the neurons in the fly brain. And you can see these little green dots here in both male and female brains. Now, in contrast, the protein that this gene makes is only made in male flies and not in females. And this difference turns out to be very significant for the difference in behavior of the two sexes here. So about the time we got into this work, my colleague Barry Dixon had made a genetic tool that revealed the structure of all of the neurons that express fruitless, so a couple thousand neurons in the central brain here. And we had the idea to test, which we did in collaboration with him, to see whether these green neurons were wired up differently between male and female brains. But the problem was that these neurons are so tangled together that we couldn't really tell whether there were differences in structure. And so what we did was to develop a sort of genetic image analysis approach to take this apart and then put it back together again in silico, so that we could then look at each of these different classes of neuron, about 60 kinds of neuron, and see whether they were different or not between the sexes. And what we found was that about a third of these neurons indeed looked different between males and females. And this likely would change the connections between the neurons in the brain. And so this is something that we went on to look at in more detail. I'm going to show you one example where we did that, which was in smell circuits. So there's various sort of names and anatomy on here, but really what you need to focus on is just the numbers, 1, 2, 3. We're going to go 3 levels through the brain. So this pheromone is going to bind to some sensory neurons here, and it's going to excite them. And then they're going to send this information to second-order neurons and then on to third-order neurons. So this is a sort of frontal view of a brain, and we're now going to see some of these neurons. So the sensory neurons send information in here, and it arrives on the so-called dendrites of the second-order neurons in yellow. They're electrically excited, and the information travels down the axon until here, this output domain. And then in the output domain, it meets the dendrites, so the input part, of another set of neurons, and we call these 3A. So they are the third-order neurons, but a particular type of them. Now, this is the situation in male brains, and this is the situation in female brains. So, you know, the color changed, but you might not immediately see that there's a big difference, but there is actually. So here, this little yellow axon is popping out because it no longer sits on top of the dendrites of these third-order neurons. So basically, there cannot be a connection from here to here in the female brain, whereas it looked like there was a connection in the male brain. So we can sort of schematize that cartoon fashion here. So basically, first and second-order neurons, which look the same between the two sexes, and then here, a connection which is being made to these neurons, which we call 3A in a male brain, but which is not found in a female brain. And intriguingly, we found another set of neurons that had exactly the opposite property. That is that they were connected in females, but not in males. Now, all of this was just anatomy. We didn't know if there were really functional connections between the neurons. And we set out to test that initially by recording from identified neurons in flies while we puffed odors onto them. The work actually of Johnny Cole, a former student. So you've got these sort of little cartoons to keep you organized while I walk through a couple minutes of data. But here is one of these neurons that we recorded. You can see that it doesn't really overlap there. And when we puffed a range of different odors at this neuron and looked to see whether it spiked, so that's these little tick marks here, it wasn't interested. It didn't really respond to anything. Now, we then went and recorded the similar class of neuron, but now in males. And when we look there, we see lots of little ticks here. And this indicated that this neuron responded strongly to CVA pheromone, but not to some of the other odors that we tested. So this confirmed, these little cartoon diagrams of what we thought the connectivity might be. We also looked at these 3B neurons that I mentioned, and what we found there was that indeed these neurons were pheromone responsive in females, but not males. So we were quite excited at this point. There were a variety of other experiments that we did, but they were quite conclusive. And this is the first time that anyone had found a sort of cellular resolution switch in connectivity inside a complex brain like this. So one of the experiments that we went on to do was inspired by this work. So this is a pretty, again, another pretty remarkable experiment in the field, done in this case by Barry Dixon, in which he was able to genetically engineer a female fly here so that it expressed the fruitless gene in the pattern that would normally be found in male flies. So here you can see this red fruitless protein, which is normally only found in males, but is present in this transformed female. She looks female, but when you see what happens next, she behaves male. So she's chasing after this wild-type female, sticking out her wing and singing. She doesn't do all of the behaviors of courtship, but she does many of them. So we went into brains of these flies, and we looked at what was happening to the circuit that we've been delineating. And what we found was that, for example, when we went and recorded from 3A neurons, which should be pheromone responsive in male flies, like this one here, but not in females, in this transformed female, indeed we found pheromone responses. So indeed the wiring pattern had changed under the control of this gene. So I'm going to summarize this set of results in what I hope is a pretty accessible fashion. Basically, we have pheromone arriving here. It's going to activate first-order olfactory, second-order, and then third-order neurons. And in the male brain, it's this set of neurons that get activated, and we imagine that they're going to take the signal off this way and trigger this rejection behavior. Now, this particular sort of switch here is genetically controlled by this fruitless gene. And when fruitless isn't there, the situation normally in a female brain, then the wiring is different. So you can see this element has stepped in place, and so then the signal goes the other way, and we think this is going to trigger acceptance behavior, promote mating by the female. So just laid out like this, there are a few questions you might want to ask. What are the inputs to this neuron that looks like it's just hanging out there, not getting any information? And of course, that's not true. And these neurons, do they actually make females want to mate? That's what we're predicting, but we actually had to go ahead and prove that. And so I'm going to give you a little update from recent results of the lab where we do just that. And that's based on this new and very important approach of optogenetics, which incidentally Gero has pioneered, in which we take a light-sensitive ion channel and we cause it, using molecular biology, to be expressed in this particular set of neurons inside the brain and no others. We can then take flies, so female flies in this case, in which this light-sensitive ion channel has been expressed in these neurons, and put them in this sort of automated behavioral apparatus where we're watching to see what they do. So here's a male fly sort of displaying to this female. And quite remarkably, when we turn on the red lights, activate this ion channel, turn on these neurons, what we find is that it makes the female more likely to accept the male, so about 40% more likely. So indeed, these neurons do actually change the female's behavioral decision when it comes to mating. So another thing that we've been doing is using an approach called connectomics to try and define the other circuit elements here. So here are these 3B neurons that I just mentioned. We've been able to go another step further, fourth-order neurons, fifth-order neurons, even sixth-order neurons, where we bumped into some colleagues. And we've been able to, again, delineate these different steps, changing the activity, either turning up or turning down the neurons. And each of them changes the female receptivity behavior. So we're piecing together a circuit here and getting slowly closer and closer to the output end, eventually, the motor behavior of the fly. Now, we found something quite intriguing here, sort of for the experts at least. These set of connections, 3, 4, and 5 here, are actually axo-axonic connections. That means the axons, the output domains of the neurons, are connecting to each other, and that's fairly unusual. But we think that it might actually be a good way to try and sequentially integrate these different pieces of sensory information in order to make a more reliable behavioral decision. So, connectomics, what's that? Well, this is actually another way in which we touched a little bit on Sidney Brenner, who was mentioned in Gary's introduction. So he was working actually at my institute, the Laboratory of Molecular Biology in Cambridge, and he decided after working on viruses and bacteria that it was time to work on something more complicated. And he chose this, the nematode worm C. elegans. So complicated is relative, you know, it's 1 millimeter long, it has about 1,000 cells, but it's actually turned out to be a gold mine. And one of the things that he decided to do was to try and find the structure of all of the individual neurons, there are 302, and all of their synaptic connections. And that was a sort of 2-decade-long project, pretty much. And how did they do it? Well, basically, they took the worm and they salami sliced it into very thin sections, and then they took electron micrographs of each section. And then somebody traced each individual neuron from one section to the next, joining them up. And it was literally done like this with pens and, and ink. So mostly it was done by this research assistant, Eileen Southgate, and eventually she compiled it into this thing that they called the mind of a worm. She also took early retirement when she was done. So it was published here in the Philosophical Transactions of the Royal Society of London, Series B, in a paper that's about 300 pages long. I'm sure it's the longest paper they've ever published. And to be honest, not much happened in connectomics for the next 25 years. So that's changed in the last 5 years or so, and that's because of signal changes in technology. So what I'm going to show you now is a new electron microscope data set that was acquired by my colleague and collaborator, Davi Bock, and a large team working with him. So let's fly into the head of the fly. Here's the brain just emerging, so about 600 microns across. So this brain was cut into 7,000 sections by Rick Fetter, and then Davi, together with a sort of engineering group, built a specialized microscope that could image all of these 7,000 sections tiled along here. So here you can see the electron microscope data. We're flying into that now, and as we get closer and closer, you can start to see more and more features of the cells. So each of these little circles is the axon, so the output domain of one of these neurons. And we're actually in an area, for people who would like to know this, called the mushroom body. So this data set is incredibly powerful because you can roam at will through the brain of the fly, find any of the neurons that you want to learn about, and see their structure and connectivity. And that's what we're doing here, using a computerized web reconstruction tool developed by my new colleague, Albert Cardona. So on this side, you have actually a synapse. So this is one neuron, and it's making connections to lots of downstream neurons here. Now, the other thing that you have is this is the whole of that neuron, this neuron shown in yellow. And what's amazing about both the fly brain and this data set is I can look at this and I can say, I know what that neuron is. It's actually a neuron that responds to CVA pheromone, one of the neurons I showed you earlier. And that's very powerful because we can then go and look who is that neuron connected to. Now, the thing is, there's an awful lot of neuron inside the fly brain. So the fly brain is tiny, as I said, smaller than a poppy seed, but there's more than 100 meters of neuronal wiring packed into that tiny structure. And when we started, we were tracing these things by hand, effectively. It may be computerized, but it essentially click. And that was taking about 1 millimeter a day. But still, we've gone an amazing distance with that through the support of various institutions and also Howard Hughes, and here in the UK, the Wellcome Trust. And so now we've been able to reconstruct about 10% of the fly brain in this way, and we focused on areas that we thought were particularly high value to understand interesting behaviors. Now, in the last year or so, computers have started to get really good at tracing, but even so, we still need human experts to fix their work and to analyze the results, and that's still a big job. Now, what are we doing with these kinds of data? So I'm now going to move on to the second and final scientific story. And basically what we've been working on is olfactory preferences, both innate and learned. So when the fly emerges as an adult, it actually encounters a complex world, but it has opinions about these things in the world. So for example, this fly thinks that fruit is good, that this toxic mold is bad, okay? And that is genetically encoded at the end through genes that instruct the development of the nervous system in particular ways. Now, I need to tell you a little bit more about the olfactory system to take you through this. But so here we're out in the periphery, the antenna, and these little colored circles correspond to about 50 different odorant receptors, each of which binds to slightly different sets of smelly molecules. So their axons project into the brain here to this structure called the antennal lobe, and they sort out. So in the end, you get 50 bumps, or to be precise, we now know 51, which each correspond to different kinds of olfactory information. And then that is then sent on to higher brain areas. So this one has been studied a lot. I've mentioned it briefly already. It's called the mushroom body, and it's required for learning and memory. Lots of good evidence for that, including from my colleague Scott Waddle. This other area has been less studied, but recently, particularly led by a postdoc in the group, Shahar Frekter, we've been able to show that there is a stereotyped odor representation in that area. Basically, what that means is you go from one fly to the next, you can always find the same neurons in here responding to the same odors. And if you think about that, that's quite consistent with a role for this area in innate behavior, because basically, if you smell, for example, pheromone, then there's going to be a neuron in here that's always going to respond to pheromone and not to mint. So flies are not dumb, though. They can also learn. And the classical paradigm here is you take an attractive or a neutral odor, maybe, and you pair it with an electric shock. And so in future, the fly will have learned that this is bad news, and it will try and avoid this odor. Now, there's actually something interesting that this made us think about, though, because let's say we have an attractive odor here, and the information coming through the lateral horn is going to tell the fly, this is good, maybe you want to go and investigate this. However, if the fly has learned that this odor comes with an electric shock, then this leg of the olfactory pathway might be telling it, careful, go away, you don't want to approach this. And so there's potentially a conflict between those two signals, and we were curious to figure out how that might actually be resolved in the brain. And so this was work started particularly by a student, Mike Dolan, that was published about a year ago. So I need to show you a little bit more of how things look inside the brain. Olfactory information comes in here, goes to these blue structures, the antennal lobe, and then comes out to the lateral horn and the mushroom body that I just mentioned. Now, what I'm showing you here in green, so is a micrograph of a mushroom body output neuron, or memory output neuron, okay? So this is a neuron that basically is involved in reading out memories once they've been formed. After learning, something changes somewhere in this circuit, and we'll see where that is shortly, and this produces an avoidance memory. Now, if you silence this neuron, you stop it working, then the flies can no longer show this avoidance memory. We were intrigued by the fact that this neuron makes a little projection into this area called the lateral horn, and we wondered if that was actually significant for its function. So we found some partner neurons that were sitting in the right place in order to connect to this neuron. And when we silenced them in collaboration with Pierre-Yves Placé and Thomas Préat in Paris, we found that the flies no longer avoided the odor that they'd learned about. So this was actually quite striking, because everything I told you before suggests that the lateral horn is supposed to be involved in innate behavior. But here it has a function in memory. So we went into the electron microscope data, and we in this case was Alex Bates, and we found this memory output neuron. And we also found these white candidate partner neurons. And when we looked in the EM data, we could see that they're actually connected. So these little green dots are synaptic connections between these neurons. So far, so good. Now, leveraging the EM data, we could also ask another question, which is who else is talking to these neurons? So we looked at all the different olfactory channels in the brain. So we're going to paint them in different colors, which we can do because we can recognize the shape of all these neurons. So there they are coming up. And then we can go into this lateral horn area, and we can reveal all the synapses, the output connections that they're making. So there they are coming up. And then we can ask which of those are talking to our neurons of interest. And what we found when we did that was something actually really quite exciting. So these white neurons receive direct input, but not from any old olfactory channels, specifically from food odor channels that would normally be attractive for the fly. And this was exciting because we have a pathway now where we have both a learned output from the memory centers and a direct innate olfactory pathway converging on one set of neurons. Okay, two streams of information coming together on one set of neurons. So we can schematize that like this. Basically, we have here this direct or innate pathway bringing in food, odor, smells, and then this learned pathway. And we think that they sum together to make an attractive response, and that we have data for that. Now, let me try and show you how this might actually let the fly solve a behavioral problem that we give it. So it's pretty common to give a sort of choice assay to figure out whether a fly has learned something. So you do something like this. You've got two odors, and let's imagine that the fly is sitting over here. And it's a naive fly. You haven't, no shocks so far. So what we think is that the two legs of this pathway, this innate or direct pathway, and this learned pathway are both active in the brain of the fly. And you get a certain amount of attraction. Now, if the fly goes over to this arm and experiences this odor, we get exactly the same thing. And in fact, we set things up so that they're roughly balanced. So that means basically that the fly has no strong preference about one arm or the other. Now, what we do next is aversive training. So we take one of the odors, A here, and we pair it with an electric shock. And there's a consequence of that in the brain, and it's this. It's that the amount of signal going through the learned pathway actually goes down. This results in less attractive signal coming out here. And this produces an imbalance, right? Because nothing has changed when the fly is on this side. And so the result of that is the fly moves away from this odor and towards that one. And that's basically how we think it solves this behavior using this circuit. So I'm going to try and do something now to give you a flavor of what might actually be going on inside the fly's head when it's doing this. So we're going to, again, zoom into the brain of the fly. I'm going to show you the different elements that are going, that are active. So here are the green neurons. Olfactory information comes in, it excites them. Boom. They actually turn on the white neurons that you just saw there, and the red neurons, which turn on a set of purple neurons, which turn on white neurons. Okay, so maybe that was kind of quick. So let's break that down a little bit. So there's an extra circuit element, which I didn't tell you about yet, but basically what we have, smell arrives here. There's a little race, and this pathway, which is our lateral horn neurons that are the final output, that gets turned on. But also this red pathway gets turned on. And the red pathway then turns on the purple pathway, which then talks back to the white neurons. So basically, you get two flashes of white neurons. And the white neurons are what are telling you, I want to approach this. Okay, now, that's what's happening over here, and it's actually the situation before learning in the naive animal. We're going to step it up a bit, and we're going to look at what would be happening after learning. And so what we think is happening there is that this neuron fires, just as before, and then this neuron fires, great, but the signal stops here at the red neurons, and it doesn't go past because there's been a synaptic change here, a depression, so that this connection has become weaker. So let's look at what that looks like. Okay, so one flash and two flashes. So I'm going to play that for you again. So you've got a little key here, white neuron fires, and then nothing happens. And over here, white neuron fires, memory neuron fires, white neuron fires again. One flash, purple, second flash. So that, in a very simple summary, is the kind of thing that we think is happening in the fly brain between the before learning situation here and the after learning situation. So we can get that one more time. Boom, boom. Okay, so it's all very quick. This is probably about, 5 or 10 times life speed. But if you want to summarize that really simply, after aversive learning, the fly thinks it's smelling less of something nice. And that essentially is the mechanism that we found. Now, this is not surprising. People have thought that for quite a long time. But what is different is we've been able to pin down exactly where these things might be happening. So I just want to take a couple minutes now to consider the benefits of this particular circuit layout. So one thing that we see here is that there's early convergence from this mushroom body area that is where the memory is forming and this lateral horn area. And we think that that's good because you could have two completely separate streams of neural processing. But if you did that, then you would have more neuronal hardware required to produce the behavior that you need to do. So this sort of seems to make sense. It also provides an opportunity to integrate the learned and innate sensory representations, because if you remember, I had that little cartoon of a fly that was kind of wondering, what should I do? Do I like this? Do I not like this? But here, this provides a way for these two signals to trade off directly against each other. And then the third thing, which is a little bit more specific, but I think a little bit more tricky, but I think actually quite interesting, is that I think it allows you to take a multimodal memory neuron and let it produce a modality-specific behavior. So what do I mean by that? Basically, what I mean is that this memory neuron can actually be involved in both smell and taste learning. Now imagine, maybe you have a fly that's learned about a bad smell and a bad taste. And then it goes into, a little bit later, it goes into a new environment, and this neuron starts doing something different. How does it know whether it's experiencing right now a bad smell or a bad taste? Because you want to do different things. If it's a bad taste, you might want to stop feeding rather than run away. So what we think is that because the behavioral output actually depends on a cooperation between the memory output neuron and this innate sensory pathway, you'll only get a behavioral response in this case, the smell pathway, because that's being activated, this sort of innate smell pathway. And so this is a way to make neural circuits more flexible, use, again, less hardware to do the job that you need. So I'm going to close with a couple of thoughts now about sort of lessons that we take away from this work more generally. So the connectomics work I already mentioned is being supported by the Wellcome Trust. And when we went for interview there, a very senior neuroscientist on the panel basically asked us, how can you learn about memory from one dead brain? That doesn't really make sense. And I mean, I know that they were testing us because they gave us the money afterwards. But I think, I thought of we gave a reasonable answer, but I gave I thought of something much better the moment I walked out of the room. Funny how that always happens. And I think there's a good analogy, which is an analogy of a tape and a tape recorder. So if you think about the complexity of what's going on there, the tape is great, obviously. It stores the song, whatever that might be, whether it's, Beethoven or the Beatles. But the tape recorder actually has much more complexity when you actually look at what's involved to make that. And it's also the same regardless of the song. And I think that's sort of the situation with the fly brain, that there's, there are areas where things change, where there's plasticity, but there's a core that is highly conserved from animal to animal, and that's what we can reveal with this kind of work. We're really trying to push ahead now, because one of the other things that I think we learn from this kind of work is in order to understand memory, you really need to understand the circuits around it. And that includes those circuits that we might think of as relating to innate behaviors, because those are what are going to be used to express the memory. And it's hard to understand the memory without having those as well. And so we're really excited to try and push into complete connectomes now, where we reveal all of the connections, not just 10%, but everything inside whole, for example, male nervous systems. Now, the final thing that I'd just like to comment on is that there's a pretty active debate at the moment in artificial intelligence about how to build neural networks. And so some people think that you should just take your neural networks, throw data at them, and let them wire themselves up without any structure. But there are others who argue that if you look in biology, brains don't just wire up completely random. It is not a tabula rasa. There are highly conserved features of brain organization all the way from flies to humans. And those have evolved over hundreds of millions of years with huge amounts of energy being funneled into them and being encoded in the genome. And so, again, in the artificial intelligence context, people think, are there learning structures that we need to put together in order to help our systems learn? And I think thinking about that tension is a really interesting mirror of the sort of work that we're looking at here. So I'd like to finish again with thanks, obviously. So I tried to mention people as I went along, but obviously, as I said, it's impossible to mention everyone who contributed. But I would like to pick out here, this is my lab at the LMB. They're an amazing bunch of creative, talented, funny people. I love going in the morning. So For the first part of the talk, so that's the sort of love part, I'd particularly pick out Johnny Cole, Aaron Ostrovsky, Sebastian Cachero, and Shahar Frekter. And then more recent work on that from Dana, Ishvan, and Erica here. The connectomics work involves particularly Marta Costa and Philip Schlegel in the University of Cambridge Connectomics Group, and also Alex Bates and Ishvan Tace in the lab. And then I'd also thank our fellow PIs, David Bock, Jerry Rubin at Janelia, Scott Waddle, and Matthias Landgraf here. The memory part of the talk, I particularly thank Mike Dolan and Alex Bates and our collaborators in Paris, Pierre Placé especially, and again, there was a connectomic input there. Now, science doesn't happen in a vacuum. I'm incredibly grateful to the support of the MRC through the Laboratory of Molecular Biology for our work over the years, and more recently to the University of Cambridge Zoology Department, who've been hosting this connectomics effort, and other institutions which I've visited or studied at over the years. The funding came from the Medical Research Council, the ERC, the European Research Council, and the Wellcome Trust, and we're extremely grateful for them. And other institutions have also given funding over the years. I'd like to finish by thanking my family, all of whom could be here today. I'm very grateful for that, for all of their love and encouragement over the years. And finally, thank you for coming and for giving, I think, about 1 penny per head to fund this work through your taxes. Any questions?
Thank you for this beautiful lecture, Greg. I think we have time for about 10 minutes of questions from the audience, which I'm sure Greg will be delighted to answer. I should mention that whoever has a question to ask should wait until they have the microphone because of the webcast. Anybody? There's one.
Hi, thank you so much for your lecture. It was really fascinating. It might be a little bit of an involved question, but the way you guys went about hypothesizing how it was that the fly kind of thought about it at the end of the, by kind of examining these mechanisms and going through it very logically, that it kind of thinks that it smells less of whatever it is that it likes. In terms of actually then testing that hypothesis, would it mean that, for instance, upping the concentration of whatever it is that it's supposed to be smelling would kind of counteract that, and then by checking those concentrations you'd be able to see kind of the comparison between how much, like a function of how much the learned versus the innate behaviors actually change in plasticity?
So, I mean, I think it's really nice to try and think about what do they think. Obviously, it's one of the awkward features of working with a fly that you can't ask it. What you can try and do is test your model, and the kind of suggestions that you're making are indeed good ones. Now, one of the problems that you always find is, let's say you do something like change a concentration. It actually turns out that that changes the set of neural elements that are engaged, right? When you have a lower concentration, you have 1 or 2 olfactory channels active. When you have a higher one, you might have 10. And all of these details end up mattering. So we can probe things like that, but I think it is quite an extended process. Right now, it's actually easier to sort of find the circuits and see if the neurons matter than perhaps in some sense build a really holistic model of what you would say the fly thinks.
Any other? Yes, there's one in the back.
Thank you for a very interesting talk, Greg. This is a bit of an extension on the previous question. Is it possible to alter, attenuate, or diminish an innate behavior by habituation?
So that's an interesting one. And I think all this, the way I would view this kind of learning is indeed that it's being layered on top of innate behaviors. Now, the odors that we use in this case are actually not attractive ones. They're mildly aversive. And what we're doing is taking one odor from being very mildly aversive to actually being quite strikingly aversive. We haven't actually tested the thought experiment that I suggested at the beginning, which is to change what we call the valence. So basically change something nice into something nasty. And actually there are reasons that we think that might actually be harder, but we haven't yet been able to probe that in detail. So I think, for this particular system, we're not sure about the answer to that question. Can you really change the meaning of something? But yes, you can take, a behavior that you would consider instinctive and then layer a learn response on top of that.
There's one in the back.
Yeah, I'm sorry if this is a naive question, but I'm not a fly person. But do you imagine the changes that you're seeing, the behavioural changes, are, again, it's not knowing the time span involved in these behavioural changes, are they something related to LTP, LDP changes or physical plasticity?
So I didn't go into it, but flies can show memory which has been characterized as different memory phases. So a long time for a fly to remember something is a day, okay? Now, the changes in the brain between remembering something 5 minutes after you learnt it and a day after you learnt it are quite distinct. But the neurons, the circuit that I've been showing about, seems to be involved in both. And if you're interested, you should actually ask my colleague Scott Waddle, who's sitting there in the third row, who is one of the great experts in this particular area.
Hello. So I'm a non-scientist, an educationist,
Welcome.
and I have a question to ask you about the pheromones. And in terms of, in animals, that creates the attraction, and to what extent in humans is this masked by perfumes?
So I guess there are always these little advertisements in the back of some of those, scientific journals, right? I remember they used to be at the back of Scientific American for years. So, I mean, the stock response there is that in mammals, there's a specialized structure that's normally involved in responding to pheromones, the vomeronasal organ, and it's vestigial in humans. It doesn't exist. I guess there has been some work recently where it seems that certain pheromones can actually be detected by the main olfactory system, which is what's at the top of your nose. So I guess, that does sort of set the clock a little bit back on that. But certainly, to my knowledge, there is no compelling evidence in the scientific literature that those kinds of human pheromones have any impact on our behavior. And just in case anyone was wondering, the fact that you can get changes in the behavior of flies in these kinds of setups, even there, we have to focus their attention on certain things. So, for example, if we want to test the impact of pheromone, we turn the lights down so that they don't have such a good opportunity to see their potential partner.
There's one up.
I'm just thinking about the metaphor of using the circuit. So like in an electric circuit, 100% it's always going to create something. Whereas with the flies, you get sort of a range, or I think you said 40 or 50%, it moves off this way. Mm-hmm. Can you say why the other 40%, why it doesn't change in the other 40 to 50%? Is it because we haven't understood the circuit well enough, or is there something else that can't?
Were you referring to the experiment where Dana Galili, who I can see in the front row, was activating these 3B neurons, and it was making the females more likely to mate? Is it that? Yeah. Okay. Yeah. So why don't they all change their behavior? So I mean, that is a good question. I think it applies just as well to flies as to humans. They're individuals, their brains are different, and we see that actually. So it's one of the interesting things that we've seen recently from our connectomics data, that if you look at the left side of one brain and the right side of one brain, they actually are significantly different. So you can see, changes in maybe 10% of the cell types might have a different number of neurons. And in fact, the difference that we've seen there from left to right is as big as the difference that we see from one fly to the next fly, which to me is slightly surprising, but is suggestive of sort of, wiring variability during development. So that's certainly one possibility. But of course, it's also impossible to make things always the same. And finally, in that particular experiment, there was sort of a ceiling effect, I think, which is basically that at some point, the flies are actually already quite keen to mate in general when we introduce them. And so it's actually hard to bump things up as high as you might want.
So I was interested in the project of doing using computers to do the tracing, but you said that you were getting traces to about 10% of the brain, and you required experts. How good are computers at tracing right now, and what do they get wrong? And given that given that experts can currently help, what are the prospects for improving them in the short future?
I mean, the prospects are very good. There's now excellent data sets available. The one that we're working with is very powerful, but for computerized learning, there's some indications that data sets which are acquired isotropically, that is where the X and the Y resolution are the same as the Z resolution between slices, can be helpful. And so we have been working with colleagues at Janelia on data of that sort, where the automated reconstruction has worked better. But I don't want to pretend that computers are the answer to everything there at the moment, because it's still taken an awful lot of expert proofreading to look at the mistakes that have been made. And essentially, yeah.
What sort of proofreading do you need?
So you need a human that looks at this 3D reconstructed neuron and says, that looks weird. And somehow, still, humans are better at that. So I'm sure they don't have to be better at that, but it's one of the things that we're taking from these data is to try and train models where the computers normally have been looking at a very small region, 500 nanometers of EM data and making decisions, but also to try and zoom out and look at the whole neuron and see when something went wrong, because that's something that the humans definitely are better at the moment. So I hope that's part of an answer.
I think we have time for one more question. Yes, in the very back.
Yeah. So since the fly's brain is very small, and how do you know which neuron does what? Because you can't really put them on the brain scanner from what I know of. So how do you do that?
That's true. So if you were studying brains in the traditional MRA brain scanner, our brain would be 1 pixel. So it's not a very useful tool. The best ways, the ways that we've used most to do electrophysiology, where you take a very thin glass pipette and you suck onto one cell and you record its electrical activity. You can also use very powerful calcium, or now voltage indicators, where you use molecular biology to put sensors into the neurons and then watch in real time how their activity is changing. And so those are great ways to do that.
So we are at the end of tonight's event, but not quite. I hope you all join me in thanking Greg for a beautiful lecture and congratulating him on the receipt of this award, the Francis Crick Medal.
Hard to get out. Okay. Thank you. Thank you very much.
And before I say goodbye, I have been asked to remind you to please complete the online feedback system. It's a trial to reduce the use of paper, and it takes just 2 minutes. Thank you.
Thanks for your positive responses.